All right, let's go ahead and go through these quiz questions so that everybody's on the same page before we jump into the lecture. So starting with question 1, what is the amino acid GLU standing for? Does anyone want to volunteer? Glutamate, exactly. Yeah, the others are not amino acids. Okay, question 2. Where would you most expect to find the hydrophobic residues in a tertiary structure? On the outer surface, inner folds regions, or both? Yeah. Exactly, yes. Yeah, so those are going to be shielded from the aqueous environment, just like what's happening in sickle cell. And 3, the protein falls into its tertiary structure because it is in the most energetically favorable conformation. True, yes. Nice. All right, quaternary structure involves multiple genes, multiple subunits, multiple tertiary structures, or all of the above. Yes, exactly, yeah. Crushed it. All right, question 5. I know this gets into a little bit of a trickier one. So we're trying to determine which side is the N terminus of this segment of a polypeptide. Does anyone want to shoot out an answer? Left, exactly. All right. Question 6. How many tertiary structures are in the following protein? 1, nice. Glad you guys got it. Nice job. All right, and then the last question, a protein with an isoelectric point of 4.5 must have more hydrophobic residues, only polar residues, fewer aspartate and glutamate residues than lysine, arginine, and histidine, more aspartate and glutamine residues than lysine, arginine, histidine, or none of these answers. More, yes. All right, good job, everyone. It seems like a lot of people are happy about how they did on the quiz, which is great. Your studying's paid off. Yeah.
So, I just want to say one thing real quick. So, again, this is quiz. If you didn't do as well as you would hope, that's okay. Again, I took this class 15 years ago, and I'm not going to say my first school was my first, but I was not whooping and cheering at the end of the first quiz. So the whole focus of these questions again is to get you to think deeply. There's going to be maybe a couple memorization questions, but the majority, each quiz, the majority of the questions will be getting you to think deeply. For example, for the tertiary structure one, the goal is to get you to think deeply about what actually is represented within the tertiary structure. And so, again, this is just the first quiz. There's going to be a lot more. And there's also the exam where you can try and beat your average quiz scores and do better. So, again, it's just the beginning. It's the first week of classes. We have a long way to go, and you guys are all going to be great.
Yeah. Thank you for that clarification. Definitely a good point. Okay, so jumping into today's lecture material, we're going to focus today on cell membranes, and lupus will be our disease topic. So a couple of the learning objectives for today, we're going to review the basic cell structure in terms of what the composition of the cell membrane is, a couple of these structural components and their composition, and then some of the pathogenesis of lupus. So this is another blue term. We talked about etiology in the first lecture, and pathogenesis is similar, but slightly different. As a reminder, etiology is the one main cause of the disease. Pathogenesis is more how the disease progresses as a result of that cause. And we'll talk about this in regards to lupus. So lupus is defined as an autoimmune disease that is associated with antibodies against nuclear proteins. So does anyone want to volunteer what they think autoimmune means? Yes.
Your immune system is fighting itself or some part of your body.
Exactly. And your immune system is fighting yourself, part of your own body, which is definitely the system, and we'll talk more about the details of that when we go into our lectures on the immune system. And these proteins, like we talked about in the first lecture, these are different structures of amino acids, but these are specifically housed within the membrane of the nucleus. So a couple symptoms of lupus: fatigue and joint pain. Photosensitivity is a big symptom, and this condition called a malar rash. So that's what's shown in this image here. It's kind of characteristic butterfly rash over the cheeks, as well as anemia and other symptoms. So this disease is commonly referred to as the great imitator because there's many different types of symptoms that can present with lupus or SLE. And so it's difficult to diagnose because there's a lot of other things that these symptoms could also be pointing towards. So the presence of antibodies against nuclear proteins is a main diagnostic criteria. This malar rash and photosensitivity are often also, but there's generally just, if you meet over a certain number of criteria, you would be considered to be a patient with SLE. So this disease specifically has different periods of remission or flare-ups. So this is when the disease is more active or less active. So the less active periods would be these times of remission. And often this is occurring due to some sort of trigger, some environmental trigger. So light, for example, is going to trigger this photosensitivity. If you're avoiding sunlight, then some of these symptoms will not present themselves as often. Any questions about this? Yes. So we'll talk about this a little bit in the next slide, but there is thought to be some genetic components, some environmental components that are playing together. It's not as directly hereditary as sickle cell, like we talked about. Specifically, it's difficult to trace exactly what causes this disease. Sickle cell was a monovalent genetic disease. Like I said, that's pretty rare. There's not a lot of diseases that are monovalent. Lupus is likely a polyvalent disease. So there's lots of genes that are going to be implicated, and that's not fully understood. Good question. Thank you. So into the prevalence and incidence of lupus. So about 1.5 million people in the US and 5 million worldwide have SLE. So this is a large prevalence. And in terms of the incidence, women are 9 to 10 times more likely to develop lupus than specifically non-Caucasian women are 2 to 3 times more likely than that. So there's definitely some types of specific populations that are more likely to maybe incur these genetic disease. Yes?
In general, more likely to get autoimmune diseases?
That's a good point. Yes, that's true. I'm sure there might be something playing into that in terms of SLE. Yeah, thanks for that point. So another way that we can talk about diseases is in terms of the economics associated with them. So in terms of lupus, there's $18 billion direct expenditures that come at the cost of this disease. So these direct expenditures are characterized by treatments, other kinds of hospital visits, medications. There's also, you can think of indirect expenditures that are associated with a disease, and that's more a function of the loss of work, the personal impacts, other things that surround the life of a person with this disease, and the economics that follow that. Yes, yeah. Yeah, that's a pretty high number. All right, like I mentioned, the etiology or the specific cause of SLE is not known. So there's likely genetic and environmental factors that are playing together. This is kind of what's thought of as what is causing the triggers or the periods of flare-ups and remissions, this exposure to these environmental factors, but that this isn't actually what's causing the disease, that there's an underlying genetic condition that is causing these kinds of unusual responses. In terms of the pathogenesis or how this disease develops, this is not fully understood. So we'll talk about one potential mechanism that this disease is progressing through at the end of the lecture today, but there's likely many other things involved. All right, so we'll go ahead and jump in now to talking about the membrane. So does anyone have any questions first before I start about the procedure? Nope. Okay, we'll get going then. So the cell membrane and many other membranes that are composed within the cell are made up of a lipid bilayer. We'll talk about exactly what that looks like throughout the course of the slides. But cell membrane is more than a bilayer. So if you just have a lipid bilayer, that's not enough to make up a cell membrane. There's lots of different phospholipids, other internal components such as cholesterol, and there's tons of proteins associated with the cell membrane. So all of these things work together in a coordinated fashion to produce the functions of the cell membrane, and there's many functions. So to start, the lipid bilayer of phospholipids have many different varieties. So the structure of these phospholipids kind of all follow the same general pattern or template. So let's get this laser pointer. So we have a hydrophobic tail that's made up of these chains, these long carbon chains. And these chains can be different lengths. They can have double bonds or no double bonds, and we'll talk more about that later in the slides. And these are connected to a glycerol. The 3-carbon molecule right here is glycerol, and this is what it looks like on its own. So you can see that where these double bonds are is where 2 of the fatty acid chains are going to be attached. The 3rd carbon on glycerol is going to have this side group connected. It's going to have a phosphate, and then on the other side of that phosphate is going to be an alcohol. And this alcohol can vary. Sorry, a lot of different animations on the side. Okay, so without the alcohol, with just the glycerol, the hydrophobic tail, and the phosphatidic acid. And this is kind of a starting point for building these phospholipids, like I said, sub out. And this can be a variety of different alcohols. So these are the 4 main alcohols that are going to be composed of in the lipid bilayer. So choline, serine, ethylene, and nitazole. And in this example, you see choline attached to the phosphate, and this whole side of the molecule is going to be hydrophilic, where this side in yellow is hydrophobic. So if you remember from the amino acids, a lot of our simply carbon chains are in that hydrophobic region. A lot of these charged groups with oxygens and nitrogens attached are going to be in the hydrophilic group. So each of these different types of lipids are going to have, and there's a general formula for how you name these. So starting with this phosphatidic acid, the first part of this is just called phosphatidylcholine, and then you can sub in the name of the alcohol following that. So this phospholipid right here is called phosphatidylcholine, since there's a choline attached. But you can sub this for any of the alcohols that are present there. Any questions about this? I know this is a lot on one slide. Yes. Yeah, so the phosphatidic acid is made up of these acyl chains, these long carbon chains, the glycerol, and the phosphate. So it has this negative charge, phosphate, so it's an acid technically, and I'm not sure exactly why phosphatidyl is the word, but that is what you would call this, this molecule right here, phosphatidic acid. Does that make sense? All right. so yeah, this is a 6 carbon ring. Sure, that's there. Okay, so in terms of how these phospholipids form into these lipid bilayers, you can see that there's a hydrophobic interior. So just like in proteins, hydrophobic groups want to associate together so that they can repel the water, that it's not creating that shield around it. There's higher levels of disorder. That's going to happen also with these phospholipid bilayers. So the hydrophobic tails are going to associate together, where the hydrophilic head groups are going to be on the outside, so facing both the interior cytoplasm and the exterior extracellular. So just as a reminder, we talked a little bit about scales in the pre-lecture, and this phospholipid bilayer that makes up the cell membrane is generally about 4 nanometers thick. So 1 nanometer is 10 to the negative 9 meters. And this, if you imagine from the pre-lecture, is going to be way smaller than even just the cell. So this is a very, very small region. But it's going to depend, the approximation, because the overall thickness of the membrane is going to depend on the length of those acyl chains on the end of the lipids that are composing of it. So longer chains will create a thicker membrane, shorter chains, a thinner membrane, but on average, it's about 4 nanometers. Proteins that are associated with the membrane can range from 1 to 100 nanometers, so they can be way smaller or way bigger than the membrane, depending on their mass and shape, their overall function. And like we said, if you remember from the pre-lecture, the cell is about 10 microns. So if we convert this to meters, this is 3 times larger than 1 nanometer. So overall, this is about 20 to 200 microns. The human hair is about 20 to 200 microns, which is way less. So just to kind of put all of these into perspective. So in terms of what this bilayer really looks like, in red here is the hydrophilic head groups, and in blue is representing the hydrophobic tails. And so these are all kind of interspersed. They're moving around. They're kind of woven around, and it's a fairly disordered structure. So there's a lot of different components that go into that, and we'll discuss that more in detail in the following slides. So specifically, the fluidity of the membrane is really important, and it incurs a lot of the functions of the membrane. So we can describe this using something called the fluid mosaic model. And this is how we think about the membrane and how things move along the membrane. So this lipid bilayer, they can all move past each other. This depends on the packing density of these phospholipids generally, but there's a lot of movement laterally with these phospholipids along the membrane. And like I said, membranes are not just phospholipids, they're proteins. There's many different types of proteins, and these will also move laterally along the phospholipid bilayer. So a couple different types of proteins that are generally associated with the cell membrane. We have peripheral membrane proteins, and these are going to be connected to the membrane through electrostatic interactions. So they're generally on the outside of the lipid bilayer, associated with that like head groups. We're going to have lipid anchored proteins. This schematic doesn't do a great job. This should be bound to the interior here where the lipids are. And this is through covalent bonding to the lipids itself. And these play a number of roles. A lot of signaling happens through lipid membrane protein or lipid anchored proteins. And there's a lot of activity that goes on around these. Another type is integral. Any protein that is in lipid bilayer is considered an integral membrane protein, and this can be membranes that either go partly through the lipid bilayer or that go all the way through the lipid bilayer. So the ones that go all the way through the lipid bilayer are specifically referred to as membranes. So does anyone have any thoughts about what kinds of amino acids would be on the interior of these transmembrane proteins? Would they be hydrophilic or hydrophobic? Hydrophobic, yeah. Yeah, feel free to speak up or raise your hand, whichever you feel more comfortable with. So if you remember these end groups of the phospholipids, these tails are all hydrophobic. So the association with these transmembrane proteins or any integral membrane proteins, the interior is going to be hydrophobic. We'll talk more about that. So each of these types of proteins are free to diffuse all the way across this lipid bilayer laterally. So it's very fluid. This fluidity is generally dependent on temperature. And we'll talk about how this is regulated in terms of specific components of the membrane in future slides. Any questions about this? All right, so as was mentioned, the transmembrane proteins are usually going to be comprised of hydrophobic residues. And one type of secondary structure that can be really useful for this is the utility helices. So looking back at what our alpha helices look like, you can see these hydrogen bonds are happening laterally in these alpha helices, and it kind of buries all of these hydrophilic parts of the backbone, these polypeptide chains, in the interior of these alpha helices, where all of the side groups are pointing out of this helix. So if you imagine that all of these side groups are very hydrophobic, then this would sit very well within the lipid bilayer of the cell membrane. So the interior, the polar backbone of the polypeptide chain is protected with all of these hydrophobic end groups that are pointing out. And this works really well for crossing the lipid bilayer. Okay, so we're gonna do a little test. I know you guys just finished, but this is a little low stakes. So just to break out our math brains a little bit, if we know that the diameter of our cell membrane is 4 nanometers, and we know that one rotation of our alpha helices is about half a nanometer, and there's about 3.6 or 3.5 residues or side chains per single turn. We're going to do some quick back of the envelope calculations, and I want you guys to tell me approximately how many amino acids would be needed to cross, how many hydrophobic amino acids would be needed to cross the lipid bilayer. So feel free to work with your neighbor. I'll give you about a minute or two to work on this, and then I'll hear what you have to say. I'll just write out. That's pretty small. It's pretty small. I don't know if anyone can read.
Just as a reminder, I should have included this on the slide, but it's worth repeating again. The thickness of the cell membrane is 4 nanometers, and that's going to be a number you need in your calculation.
All right, I'll give you another 10, 15 seconds. Okay, who wants to volunteer their answer? Yes. Yeah, that's exactly right. Do you want to share how you got there? Yeah. Exactly. So just to write that out, for those that wanted to see it, we start with 4 nanometers. If you divide by half a nanometer per turn, that cancels out, and then multiply that by about 3.6. This gives you roughly 25. Not exactly, but we're just doing some back of the envelope calculations here. All right, does that make sense? Is there any questions about this? Okay, and that number is going to be important because we want to be able to recognize what parts of a protein are crossing this lipid bilayer. So if we're looking at a primary protein sequence, we talked about this a little bit, but how would you identify a part of a protein is membrane spanning? So we want to look for a stretch of amino acids about 25 in number that could form an alpha helix and are all hydrophobic. So this is going to be our criteria. And there's more of a mathematical way to kind of get about this. So I'll walk through this slowly. This is a hydropathy plot or a hydrophobicity plot. And so this is one way of plotting your chain of amino acids. So I'll just start here with the x-axis. So the x-axis is the position of your sequence amino acids. So if you remember when we put up the sequence of hemoglobin beta chain, it was just a string of letters. And the first one would be position 1, last one would be position 90. So here, this is about a 350 amino acid long protein. On the y here, we have either more hydrophobic or less hydrophobic. And this is actually given numerically. So there is a way that researchers can talk about the amount of hydrophobicity or hydrophilicity of an amino acid, and this is given quantitatively. So you can actually look up these tables that give a number towards how hydrophobic an amino acid residue is. So those residues are going to be used in calculation, and this calculation is kind of like a moving average. If you think about, I know we're almost to election season or already started, but if you think about like polls or other types of data where you're trying to quantify changes over time, usually those changes can be sudden. And so a lot of times what people do is they don't take the number for each day or what the polls are saying for that day. They'll take the average over 1 week. So maybe 5 days before and 5 days after. And that's a little bit more of a stable number in terms of what things are saying in the polls. And that's a little bit how like this hydropathy plot works. So because in these membrane stretching parts of a protein, doesn't matter if you just have one hydrophobic amino acid, you need a whole length of acids. So some of those are going to be more hydrophobic or less hydrophobic, but you want the average across a length of that protein to be overall hydrophobic. So above 0 would be hydrophobic, below 0 would be less hydrophobic or more hydrophilic. And if you go across the length of this protein, this little schematic kind of shows this red box in the middle would be that actual spot in the amino acid chains, and then the blue on either side would be the other residues that are taken into account in this moving average. So each of those residues has a number for how hydrophobic. Just going to average it and then plot it in this plot. Does that make sense? Is there any questions about this so far? Yes, no? Yes. Yeah, great question. So this is used for figuring out what parts of the amino acid would be stretching across the lipid bilayer. So having a stretch of amino acids where there's about 25 that are all hydrophobic is going to be a pretty unique structure for alpha helices that are crossing the lipid bilayer. So does anyone want to volunteer what parts of this might be those alpha helices that are crossing the lipid bilayer? Somebody brave. Yeah, so what part of this plot might be corresponding to the sections of this protein that would be moving across the membrane or through the lipid bilayer? Yes. Yeah, exactly. So you can see from one of amino acids that are all on the positive end of this plot. So they're all fairly hydrophobic. And this would be true about all of these stretches. Does that make sense? And 25 is kind of a rough estimate that we use for this. It could be slightly less, it could be more. You could have proteins that are crossing the lipid bilayer multiple times, but generally about 25 that are all in this positive region are going to be, yes. No, this isn't taken from direct measurements of the protein. This is taken from the sequence of the protein, which could be from chromatography of some variety. But assuming that you already know the sequence of the protein, you're just going to be looking at the table with all the hydrophobicity numbers and then taking these averages at each point. Does that make sense? Yeah.
Does it tell you that it's definitely crossing? It's just like could cross.
Yeah, it's telling you that it could. But if you look at different types of proteins, so some that you know are not going to be associated with the membrane at all, there's rarely, if ever, this structure in them. So it's usually going to be kind of closer to zero, more random between hydrophobic and hydrophilic. There's generally, it's okay to infer that if there's this long stretch of hydrophobic amino acids, and especially if you know that it is associated with the membrane, that there is going to be some lipid bilayer crossing. Good question. Yes. Yeah, so you mean when it's folded? So yeah, the primary structure can only tell you so much, right? So from it, you can only infer the actual amino acids, whether they're hydrophobic or hydrophilic. Getting at the secondary structure and especially the tertiary structure of proteins, especially as they relate to the membrane, is very difficult to do. There's different modeling softwares that you can use that kind of take into account all of the different interactions that we talked about in the last lecture and kind of predicts what the structure is going to be like. So you can't know that these are going to be alpha helices. You can't know for sure, like I was just saying, that it's going to be crossing the membrane, but you can use these kinds of measurements to reasonably infer it. So we can't know for sure that these are alpha helices. They might not be alpha helices, but if they're about 25 residues, that would be way more than you need for just a regular stretch of the protein crossing the membrane. So a lot of times this is how it's done. Or I mentioned before in the beta sheets, beta barrels are a really common type of secondary structure. So this is multiple beta sheets associated together. And this kind of creates like a channel through the membrane. So especially proteins like this that are crossing multiple times, a lot of times it is a beta barrel. And so you can kind of guess the structure, you can look at the amino acids, you can figure out what its function is by different cell experiments, and all these are clues that can get at a little bit more of where is this protein, what's its function, and what the secondary structure might be. Good question. All right. Any other questions before we move on? The main takeaway from this slide is just to know how to read the plots. If we would say what portions of this would likely be crossing the cell membrane, being able to identify that. Okay, so a lot of the membrane function is going to be dependent on the different types of proteins that are associated. So the lipid bilayer, like all of the other cell, a transport barrier that allows specialization for cells, that allows like a reducing environment in the cytosol where it's not going to be in the extracellular space. It's going to provide a lot of different energy gradients and ion gradients that allows the cells to function. But a lot of times there needs to be transport across these membranes, and proteins that are associated with it are going to provide a lot of those functions. And this is a very active site. So this is a little tidbit I got from Professor Zunder when I was TAing this class with him. He said about 30% of the functional proteins in your genome are actually membrane proteins. So that is a ton of proteins that are doing a lot of different activities at the cell membrane. Okay, so some of those functions, mineralization. This is the main function of the lipid bilayer, like I was saying, and this is going to localize some reactions. So for example, there might be some metabolic reactions that are only favorable at high concentrations of a certain reactant. And so you need to maintain those high concentrations within a compartment in order for those reactions to progress. Similarly with ion concentrations and other things. So this allows kind of your reactants, your proteins that are catalyzing those, and the necessary components to be within regulation of transport. So this, especially for larger proteins or other components that wouldn't be able to diffuse the lipid bilayer, these proteins on the membrane are going to be serving as different kinds of transporters to get things across specifically. This is very important for signaling cascades. So you can think of any kind of signal that the cell receives is generally going to be coming from something external to it. And then that signal is going to create some downstream impact in the cell itself. And that's generally because of a receptor and a ligand that's transducing a signal. So if this is your lipid bilayer, say we have a protein and there's some receptor. So a receptor is any protein that's going to bind a ligand. So a ligand, this can be any molecule that binds to a receptor. It's a little bit of a tautology, but receptors bind ligands, and that is their general definition. And then the binding of this ligand might change something conformationally in your protein. So this maybe could be phosphorylating itself, or it might attract another protein that exists as some sequence of events. Maybe that's going to come to the nucleus, and then that's going to change something about how the cell behaves. Does that make sense? Okay, so this is generally considered as a receptor cascade, so some kind of ligand binding a receptor, producing a change in a cell, and that transduces many signals. There's lots of things that happen in this arrow in between, a lot of different types of transduction pathways that can occur, but generally this is a structure for how cells are responding to their environment, and this is generally because of these membrane proteins that are able to receive these signals. All right, so cell-cell and cell substrate binding, also cell membrane through different adhesion receptors. So we'll talk about this way more in detail when we go through the ECM lecture next week. But this doesn't have to be a ligand that a receptor is binding. It could be another thing on another cell. So maybe there is another cell nearby that likes to associate. So this happens a lot like in your skin or other kinds of barriers in your organs. There's cells that need to associate really close to each other. Your blood-brain barrier is like this. The cells are associating very close to each other. And so there's actually binding that happens through membrane proteins that helps to maintain that binding between cells or between different components of the extracellular matrix that exists. And again, we'll talk about this in more detail next week. Okay, any questions about these proteins before we move on? These are just a lot of these things we'll talk about in great detail, so you'll become very aware of these, all of these functions by the end of the class. Okay, so a little bit more about membrane fluidity. The regulation of this fluidity is very important for maintaining these functions that are happening at the membrane. And this is done in a lot of ways. So like I said, this fluidity is going to be responsive to temperature. And so if it gets too cold, the crystallinity or the solid nature of these membranes will increase. And so this can be a real problem in cold-blooded animals, or if you're trying to design some kind of drug delivery that you're trying to get across the cell membrane, you want to make sure that things can diffuse, that it's not too solid, that membrane proteins can still interact with other things on the membrane like they need to. And so a main way that this can occur is the type of fatty acid chains that are composing your cell membrane. So here's a couple examples. This fatty acid chain would be considered completely saturated. So you can think of something saturated as in there's no more reactions that can occur with it. So at every carbon, there's 2 hydrogens, and there's no more reactions that can occur in that state of a molecule. But if you use a double bond, this is now considered unsaturated. So technically, something that's unsaturated can still be reacted with. Double bonds are generally reactive. These double bonds can be either cis or trans. So if you remember from chemistry, cis means same side. Actually, I think this is orgo. I don't know how many people are in orgo yet, but cis means the same side, and trans means opposite side. So in the cis double bond, it comes in from one side and it goes out the same side. In a trans double bond, it comes in from one side and it goes out the other side. Affect the structure of these fatty acid chains. So if you can see in the cis double bond, it's a little not as straight as in the trans one. And this is going to impact the fluidity of the lipid bilayer. So if you have a bunch of really straight lipids, then they can pack really closely together, and they can be more fluid. Versus if you have, sorry, I said the opposite. If there's trans double bonds, it'll be similar to the saturated fatty acid pack really closely. The cis disruption to that packing, and there's more fluidity that is happening at the cell membrane. Does this make sense? And we'll talk about this more. Yes. So if you have purely straight acid chains, whether that be saturated types of chains or through trans bonds, that's going to be more rigid. So you think about, there's a lot of space for the lipids to move by each other. It's more difficult. Does that make sense? Okay. And this double bonds occur in something like olive oil, which at room temperature is going to be liquid, and either animal fat or Crisco. I don't know if people use Crisco anymore, but If you keep it at room temperature, it's going to be solid. So this solidity comes from the packing of these fatty acids. Just maybe a way to help remember that. So another aspect that's regulating the fluidity of the membrane is cholesterol. We'll talk about that in just a few slides. Another thing that can regulate the fluidity in terms of acid chains is these kinks. So like I said, these are all trans double bonds, and this is generally what is synthesized in our cells. So our cells are always synthesizing trans double bonds, and this is essential for our cell membranes. Another fun fact I got from Eli, this arachidonic acid is essential for all of our cells. It's present in all of the cell membranes, and it's only found in animals. So plants don't make arachidonic acid with these trans double bonds. And the enzyme that facilitates this synthesis, cats don't have that. So cats are obligated to be carnivores because if they were only eating plants, they wouldn't get any arachidonic acid. So if anybody has cats at home, make sure you feed them meat. Okay, another aspect of the fatty acid chains are these omega bonds. So you might have heard of different omega-3s, omega-6s in terms of being essential to your diet. This is also because this is something that we can't synthesize, so we have to get it from our diet. And these omega bonds are cis double bonds, same side. And the number after the omega refers to where the first double bond occurs from the far end of this 1, 2, 3rd spot is this omega-3 linoleic acid, and this omega-6 arachidonic acid occurs at the 6th carbon from the end. So it's just a good thing to know this nomenclature. All right, we're going to talk about cholesterol now in terms of how this regulates membrane fluid. So this is a component of all of our membranes, and it serves as kind of a fluidity buffer. It acts in a really unique way in terms of regulating the membrane fluidity. And this is the structure of cholesterol. You don't have to be able to memorize it, but I think it's helpful in terms of describing how it's interacting with this lipid bilayer. Has all of these different ring structures and a lot of carbon groups, and this whole end of the cholesterol molecule is very hydrophobic. So this side of it is going to be inserted into the lipid bilayer, where this hydroxyl group at the end is hydrophilic. And so this sticks out. So generally, because of the size of this molecule and this hydrophilic nature, it's only going to be associated with one leaflet of the membrane, and this hydroxyl sticks out with the hydrophilic head groups. So how this works in terms of membrane fluidity regulation, when you have this cholesterol in your membrane, it kind of disrupts the packing. So similar to how these kinked double bonds disrupt the packing and they lower the fluidity of the membrane, the insertion of cholesterol into the fatty acid, the inside of the lipid bilayer, can disrupt some of this packing and increase the fluidity of the membrane. And this is specifically happening at low temperatures when the packing is high. But then at high temperatures, you have fluidity, and the cholesterol in there kind of works in the opposite way. So this is good. We want to regulate some medium amount of fluidity that is be essential for our cells to stay together, but to remain functional. And so the part of this molecule attracts all of these hydrophobic tails of the lipids in the bilayer. So it kind of sticks things together. It maintains some amount of fluidity, even at super high temperatures when the fluidity will naturally increase. Does this make sense? Any questions about this? Yes. Yeah, so temperature is about 30 degrees. I think probably degrees above that would be a high temperature, 5 degrees below. So if you, I'm sure everybody knows from experience, you don't want to have like over 105-degree fever. That's going to be starting to cause some problems in many parts of your physiology, but your cell membranes are definitely not an exception. Good question. All right, this is definitely a key concept. So if you have any further questions or just want to talk through this, definitely feel free to come to any of our office hours. Just understanding how cholesterol works at both ends of the spectrum is very important. Another role of cholesterol, it's going to be highly enriched and called lipid rafts. So lipid rafts are going to be thought of as these like structures that are all associated together. And so they'll kind of move together in the cell membrane. And lipid rafts are composed of lots of things, different raft-associated transmembrane proteins, and they're highly enriched for cholesterol. So the red here is cholesterol. You can kind of see how the end will stick out with the head groups of the lipid bilayer. And these are large structures. A lot of signaling occurs in these lipid rafts. And that's present, or that's shown here with a lot of this glycosylation. So what glycosylation is just the addition of sugar molecules. And there's different proteins that can be bound through these sugar molecules. So these are called GPI anchored proteins. A lot of this is just causing signaling between cells. So this is one of those maybe ligands that another cell could recognize. And we'll talk more about some of these things later in the class. Okay, we're gonna switch back now to talking about SLE, lupus, and the pathogenesis or potential pathogenesis. So is there any questions about what I just went through before I switch gears here? Nope. Man, you guys are on it. Cool. Okay, so going back to the structure of the phospholipids, thing important in terms of these is these alcohol groups on the head of these phospholipids are going to be specific to the inter or the outer leaflet of the cell membranes. So when they're synthesized, if you remember from the pre-lecture, they are transported through the Golgi, through secretory vesicles, and then they'll fuse with the membrane. So they'll fuse on the membrane, and then once they're on one side of the membrane, we can measure this experimentally. They don't flip to the other side of the membrane, except for very rarely. So this occurs spontaneously about once a month. So one phospholipid a month might swap between the inner and outer leaf. But what is occurring is there's a lot of regulation in terms of what side of the membrane each of these phospholipids are occurring through different transmembrane proteins. So scramblases is a general category of proteins that are going to be transporting these phospholipids from one side of the leaflet to the other. So this cell membrane is thought of as asymmetric. So there's going to be different types of head groups, of phospholipid head groups on the inside of the membrane versus the outside of the membrane. And you don't have to know which ones are where or the names of these specific enzymes, but it's important to know the asymmetry and that they're not going to move from the inner and outer leaflet on their own, except for very rarely. Okay, so in terms of SLE, this phosphatyl serine is going to be really important. So this lipid is going to be primarily present on the inside of the cell membrane. So we'll introduce something now called apoptosis. So this is a type of cell death that is very regulated. It's very planned and coordinated by your cells and can happen for lots of reasons. Maybe there's some DNA damage that is not able to be repaired, irreparable, or many other things. And this is going to cause some signaling events. And then the cell is going to look like this. So the scientific term for what's happening here is blebbing. So these little vacuoles on the outside of the cell, these are called blebs. And so this is what a cell looks like when it's going through apoptosis. And like I said, this is very regulated process of cell death. The first step in this is the recruitment of these find me signals. And there's a couple of these, but one of these is this phosphatidylserine is going to be transported to the outside of the cell membrane. So there's a specific transporter protein that is going to be upregulated in response to some trigger that's within the cell saying it's time to apoptose. And so this transporter is going to be transcribed, delivered to the cell membrane, and it's going to start transporting phosphatidylserine to the outside of the cell. And so this allows different immune cells, specifically phagocyte-based immune cells. So this just means a cell that eats another cell. And macrophages is a main one of these. And we'll talk way more about these when we get to the immune lectures. So these macrophages can, through this receptor ligand binding cascade, recognize these phosphatidylserines on the outside of the cells, and this triggers for them to engulf this whole cell. And this is a good thing for the natural regulation of your cells, for your body and the state of your organs in general. And so this is something that we want to happen. This is something that the cells have planned and is a good thing. So in lupus, there is something damaged in terms of the recognition of macrophages of this phosphatidylserine. So we don't know if that is just an inability of transporting the phosphatidylserine to the outside of the cell or the macrophages being unable to recognize it. But what that leads to is an ongoing blebbing of apoptosis. And eventually these blebs have full of cell components and they're just going to degrade. And they're going to release all of their components into the extracellular fluid, and not good. So this is not something that is normally recognized by immune cells and is going to trigger a lot of different issues. And so nuclear proteins in particular, in terms of lupus, will trigger all sorts of inflammation. So these nuclear proteins are not generally supposed to be on the outside of the cell. They can be toxic to other cells. And so the immune cells are going to recognize them and say something is very wrong and cause all sorts of inflammation. And so this has been shown and can be measured, and is one potential mechanism for how lupus is progressing. So in terms of research, if you're interested in this, Peter Casson is a professor in BME who does a lot with structural biology and membrane proteins. And Cornelius is a new professor at UPenn that goes through molecular mechanisms of cell death and is doing really cool work if you want to look into what he does. So before you guys go, a couple reminders. So the pre-lecture for the ECM next Tuesday, I will post before the end of the week. I'll get that to you guys soon, as well as the slides for that lecture. We're going to have the journal club next Tuesday. So the information about the journal club will be in those lecture slides for Tuesday's lecture. And then you should have your concept for your projects by a week from Tuesday. So just a reminder to start thinking about that and think, yes. We don't know. Yeah, good question. Yeah.